Hello, everybody. Welcome to biology class with Mr. Mustafa Abiodun Musa. So this has been revision for the two lessons we had before we stopped teaching. Some students were not in school, but this is just to let you know we completed everything on the cellular reproduction with the students in school. So I will do this video to be a reminder or a revision for the ones who were absent when the lessons were taught. When we say cellular reproduction, we're talking about reproduction that happens in the cell. Cells, they reproduce, the cells multiply, the cells replicate. All of these things are what is being understood when we say cellular reproduction. So what are the things we should understand today? The focus question will be, what are the primary stages of the cell cycle? Cell cycle refers to a cycle of the cell. What does that mean? The meaning is talking about the cell from the time the cell became alive to the time the cell died. So the meaning is a cell cycle means that the cell, right from the time it began to live, became to become a living thing, to the time it stopped to become a living thing. All of the things that happens in it is called a cycle. So the cell cycle is divided into different phases. So it says is a sequence of growth and development stages that an organism goes through during its life. So how the organism starts to grow, start to develop, or how the organisms increase in number, increase in mass, all of these are understood as part of the life cycle. So where we talked about the surface area to volume ratio, the meaning is we're talking about how an organism size would affect the surface area. We have several factors that influence the cell size. Surface area is the area covered by the plasma membrane. The plasma membrane is a membrane that covers the entirety of the cell. So when you talk about surface area, you're talking about all of the area which is covered by the plasma membrane. The plasma membrane is a structure through which all of the nutrients and the waste product would pass out of the cell. This process is called diffusion, when materials are expelled out of the cell. The volume is a space which is taken up by the inner content of the cell. If you have a cell, the inner content of the cell, the space being taken up by the inner content of the cell is called the what? Is called the it's called the volume. Let's say we have a cell like this. So all of the space here, all of the space inside the cell right here is called the what? Is called the volume. So the volume is a space being taken up, being covered by the plasma membrane. So a cell volume would be a cell, when a cell looks like this, we would say it's smaller than a cell that looks like this. So the volume of a cell differs based on the volume, that is to say the inner content and the things which is contained in the cell. So now let's look at some examples here. We have a ratio to surface area to volume ratio. We want to compare the surface area to volume. When a cell is large, the summary of the section is, when a cell is large, we say the cell has a less ratio of surface area to volume. So the surface area to volume ratio of a cell decreases as the cell gets bigger. The bigger the cell is, the less the surface area to volume ratio would be. So cells with a higher ratio to surface area to volume can sustain themselves easily. So when a cell is got a higher ratio of surface area to the volume, then we would say the cell is very, very small. So the cells can easily sustain themselves. So this is exactly what it is. Let me give an example. This is a nucleus. You can see how because the cell is small, it has the ratio, the surface area to volume ratio is very huge. And this is getting smaller, and this is very small. Now you can see the nucleus is very small compared to the general cell itself. So transportation of substances in the cell. As you all know, when a cell is very large in size, materials would move slowly within the cell. So smaller cells can transport substances more easily. When you transport things through a small cell, it gets throughout the cell very easily. But when you transport things through a massive cell, it takes a longer time for the materials to spread across the organism. A good example is when you go to the hospital to take injection. The first thing is which has been done is your weight and your age and your height is been collected, is been measured. Why do they do this? They want to know how much grams of medicine to be given to you. If I want to take, for example, I have the same disease with someone who weighs like 100 kg and my kg is 50, despite the fact that we have the same disease, they would want to give me a less dosage compared to someone who's got double my weight, because he has double my weight, he's got more the diffusion in the body would happen at a slower rate, and he would need more substances to be able to take care of his problem. So the fusion is not efficient if the distance is long. So like you press a perfume in a small room, in a classroom, you will see the smell of the perfume will be more than when you press the perfume in an auditorium, in a very big gymnasium. So this is an example of transporting substances through a cell which is massive and through another cell which is very small. So the cytoskeleton transportation network becomes less efficient for a cell if the distance to travel becomes too large. So if the cell, as you know, the cytoskeleton is a skeleton which a cell has if it does not have it doesn't have a normal skeletal system. So the cytoskeleton is just like some of the microtubules and microfilaments that makes it easy for the cells to have a shape. So they usually use all these things to move around. Also, cell communication. When cell communication is more efficient for cells to communicate if the cell is small compared to if the cell is very massive. So if you have a small cell, the communication is pretty easy compared to if the cell is very, very massive. As I was saying, so now chromatin and chromosomes. Chromatin is a relaxed form of a DNA. A DNA in a normal sense, when the cells are growing, when there is no cell division, the chromatin is very relaxed. It's a relaxed form of a DNA. So this is, I mentioned to you, if you could still remember, is very relaxed, and it looked like some threads which are just spread around in the nuclear material, in the nucleus of the cell. But when it becomes a chromosome, it becomes condensed. That is to say, the strings, they gather together more. For example, if you have, let's say you have the chromatin looks something like this, very spread out. So these are the genetic materials. But once the cell is about to divide, they will fold around, and fold around to become something which we call the DNA. So they would become they become condensed in a condensed form, in well-organized form. So this is a major difference between chromatin and chromosomes, if anyone is asking you. So they are actually more condensed and more visible because, when they become clustered and they become condensed together, you can easily see them when you check the cell under the microscope. They pass from one generation to another one because they carry the genetic materials. Eukaryotic cells, I think you know what eukaryotic cells are. They're cells with a membrane-bound organelle. The DNA is organized into what we call chromosomes. The phosphate groups in the DNA would help to create a negative charge, which would attract the DNA to the positive charge histones, histone proteins. The histone proteins, they are protein molecules that stick around the chromatin. So they stick around the chromatin, and the histones with the chromatin form what we call the nucleosome. So the nucleosome is grouping group together into chromatin fiber, which will supercoil to make the chromosome. So as I told you here, so it supercoils, it twines together. I mean, it folds around each other and become a chromosome. So what we want to derive from this section is the chromatin together with the histones from the nucleosome. And nucleosome, when it's being coiled in a proper way, then becomes what we call the chromosome. So let's talk about cell cycle in more detail now. Cells reproduce by a cycle of growing and dividing called the cell cycle. So what a cell cycle is a process by which cell grows and divides, that is to say, replicate. The cell cycle is divided into three sections. One of the sections is called the interphase. Interphase is a stage during which the cell grows. Interphase is a stage during which the cell grows, develops into mature functional cell, duplicates the DNA in the nucleus, and prepares for cell division. This is what we call the introduction or the preamble stage of cell cycle. So interphase is been known to be part of the cell where the cell would grow normally. The cell normal growth would happen. Then the genetic material, the nuclear material would increase in size because it had to double. Because when the cell's division happens, that is to say, one cell becomes two cells. So due to this reason that the genetic materials will be spread between two cells, it has to increase in size and in number. So this is what makes the nuclear material becomes larger in size. So, and it prepares for the cell division. Interphase is divided into three stages. The first one, we call it the gap one phase. Then the second one is synthesis, and the third one is the gap two phase. As I told you before, the gap was actually called the gap because the scientists thought that nothing was happening in the gap phase. But today, the scientists have decided to name it the growth phase. So what does that mean? The meaning is this is when the cell starts to grow. The normal growth happens in the cell grows a normal life. The cell taking food and they use that to grow. Then the next stage is the synthesis stage. This stage is when the chromosome would have to replicate or double because the cell is preparing itself for division. And the gap two phase is a phase in which the nucleus is ready to split into two. So these are the things that happen in the interphase. So the interphase is just the beginning of the cell cycle. So this is the repetition of what I just mentioned to you. So the gap phase, which is growing, will happen. The cell will carry out the normal cellular functions and they would prepare to replicate the DNA. So in the synthesis phase, this is when the DNA gets replicated. So it prepares for cell division. And the gap two phase is the preparing for this division of the nucleus. So the nucleus wants to divide in the gap two phase. So this is, as I mentioned to you before. So now there is a second phase. Now I want you to understand this section. I don't want it to be very confusing. Cell cycle is divided into three: interphase, mitosis, and cytokinesis. So I talked about the interphase because the interphase is the first one. Is divided into three stages: the gap one phase, the synthesis phase, and the gap two phase. Now I want to go to the mitosis phase, which is the second phase of cell cycle. So in the mitosis phase, there are several things that happen in it. So this is a stage where the cell actually divides. The cell division happens in the mitosis phase. So I was taught by talking about the phases one after the other. In your section, in your textbook, we have four phases, which is the prophase, the metaphase, the anaphase, and the telophase. So the prophase is the first and the longest stage of mitosis. What happens in prophase? the chromatin, which we talked about before, that is very loose genetic materials. They condense to become a chromosome in the prophase. Then the sister chromatid. What is a sister chromatid? If you have a chromosome like this, let's say this is a chromosome, this one is a sister chromatid, and this other one is a sister chromatid. The meaning is in one chromosome, you have two sister chromatids. So the sister chromatid are structures that contain identical copies of the DNA. So the copies of the DNA on this sister chromatid here would be the same as the DNA on the other sister chromatid. So they are being attached together at the middle here, which is called the centromere. As the prophase continues, then the spindle fiber, the centrioles, the ester fiber form a spindle apparatus, which is shown in the next slide. So in the next slide, this is exactly what happens. So as you know before, the sister chromatids, this is a chromosome, but this is sister chromatids. So the ester is a combination of the centrioles. The centrioles always form when the cell is about to divide. So it usually form together. Then this would migrate from this section to this section. So to be in the poles. Then they would produce a spindle fiber, which will spread out and connect with the centromere of the chromosome. So during that period, they try to stick themselves properly to the chromosome so that they can be able to pull the chromosomes apart from each other during the process of cell division. So now in the metaphase, as you know, during the cell division, the cell wants to divide properly. What would happen is the chromosomes would align themselves properly in the center of the cell. So the second stage of mitosis, the sister chromatids are pulled along the spindle fiber. So the spindle fiber pulls them along itself, arranging the chromosomes in the center of the cell, making them to line up. It's called the equator. So this would happen, and then the process of cell division would happen. So what would happen is the spindle fiber will get shorter. They continue to shorten themselves. As they shorten themselves, they would separate the sister chromatids from each other. So they would divide the sister chromatids away from each other, and they would migrate away from each other to form a individual sister chromatid. So this would migrate this way, and the other one here would migrate this way. So this would do the same thing. This would go here. So you would say this goes here, this goes here, this one goes here. So the same thing happens at every stage. Same thing happens at every stage. So the same thing happens here. So the same thing happens here. So this would happen. So they will separate. So when that happens, and then we call that an anaphase. The anaphase is the third phase of mitosis. Mind you, let me remind you again, we are still talking about mitosis, which is divided into four phases. We've talked about prophase, the first phase of mitosis. Now we're talking about we talked about metaphase, which is the second phase of mitosis. Now we're talking about the third phase of mitosis, which is anaphase. So what happens is the sister chromatids, they're pulled apart. The microtubules of the spindle apparatus, they begin to shorten. So the sister chromatids will separate from each other. The chromosomes move towards the pole of the cell. So then in the telophase, this is a phase where the mitosis is ending. So what happens is now you would see you had the chromosome. Now the sister chromatids have not separated into two halves, four here and four here. And now the nuclear material, which was dissolved in the beginning to allow the genetic materials to come out. So the nuclear membrane is forming around the nucleolus again. You can see the nuclear membrane is forming again. So what happens is the chromosomes arrive at the pole and begin to relax. So two new nuclear membranes begin to form, and the nucleoli would reappear. So these are the nucleoli reappearing. So now you can see you're getting two cells from one single cell. So now after the process of mitosis, then we call the third phase of cell cycle is the cytokinesis. So cytokinesis, it's towards the end of the mitosis, the cell begins cytokinesis, by which a cell cytoplasm divides. It divides, resulting in two cells with identical nuclei. During the cytokinesis in animal cell, the microfilament constrict and pinch off to form two cells. That is to say, you will see something like this if these are two cells. This is, let me show you from the other slide. So this is what we pinch in the middle here. So pinch, this is for plant animal cells. So they can pinch in the middle and separate and cut off to form two cells. So this is what happens in the animal cells. But for the plant cell, because the plant cell has a cell wall, it's not easy for the cell to pinch and separate. So instead, they form something called the cell plate. So let's say this is a cell and it's about to divide. So the cell plate forms in between the two cells, and the cell plate is being filled with cellulose materials or some sucrose, cellulose materials or sucrose. Then it hardens, it becomes harder. Then this will constrict the cell into two different cells. So this is how it happens in the plant cell. So the plant cell would not be able to pinch in the middle or constrict in the middle the way it happens in the animal cell. So they will produce what we call the cell plate. Then they would separate the cell into two different cells. So now the cell have specific instructions for carrying out the cell cycle. So this is what brings about the cycling. A cycling cell, a cell cycle in eukaryotic cells has been driven by some combination of two substances. The substances are proteins called cyclin. So the cyclin protein would bind with an enzyme called the cyclin-dependent kinases. So the cyclin-dependent kinases is called the CDKs. So the protein with the enzyme cyclin-dependent kinases, they form a kind of combination, or they form a complex. So this complex is what controls the cell division. So different cyclin CDK combination control different activities during the cell stage of the cell cycle. So this brings about brings us to the quality control section. So built-in checkpoints would monitor the cell cycle. So what happens is, before the cell cycle happens in the gap 1 phase, so the cell would check if the DNA is damaged. If the DNA is damaged, so what happens is the cell cycle would not happen. It helps to fix it. Then if it's not fixable, it destroys the cell. But if there is no problem with the DNA, then it goes to the next stage, which is synthesis. In the synthesis, this is when the DNA replication happens. So if the DNA replication is done properly, it says yes, then it goes to the gap 2 phase. So in the gap 2 phase, the DNA replication is already completed. So he wants to check if the DNA replication is completed from the synthesis. Then if that is okay, it says go to mitosis. So, and during the mitosis, the most important thing is for the spindle fiber to attach themselves properly to the chromosomes. Because if they fail to attach themselves properly to the chromosomes, they could fall off during the process of, during the prophase stage, where they're pulling them apart from each other. So this is what we need to understand. There is something we call apoptosis. Apoptosis is an organized cell death. So it's an organized cell death. So what does that mean? Not every cell is destined to survive. Some cells will go through apoptosis or programmed cell death. So the cells will shrink or shrivel in a controlled process, because if the cell is not having the genetic material being done properly, the cell will cause a problem. So the cell will be killed instead of forming an abnormal new cell. So an example of apoptosis is what happens in the human hand and the foot. So the cells in spaces between the fingers and the toes, they would undergo apoptosis, which is programmed cell death. And that would give the gap between your fingers. An example in plant is a localized death of the cell of the leaves during the autumn, when it's preparing itself for the winter. All the leaves from the trees will fall down, because the cell connecting the leaves with the branches, they would die normally. So when there is abnormal cell cycle, it leads to cancer. So cancer is an uncontrolled growth and division of the cell. Cancer results when the cells stop responding to the control of the cell cycle. So the cancer cells can kill an organism by overcrowding in a place. Maybe the organ or the tissue will be compressed. So this is how the cancer cells becomes a problem. But if the cancer cell is growing in a place where there's no disturbances to any cell organs or tissues, you would not know the person has got a cancer cell growing in the body. So what could cause a cancer to grow in the body? Mutation. Mutation causes cancer cell growth and division. Environmental factors can also affect the occurrence of cancer cells. Carcinogens, that substances or agents known to cause cancer. Not all cancer can be prevented, but avoiding known carcinogens can help to reduce the risk. What about cancer genetics? More than one change in the DNA genetic makeup is required to change an abnormal cell into a cancer cell. So when the cell is the DNA genetic material of a cell, when it changes, the cell could become cancerous. So this might this might explain why there is risk in an increase with age. If someone gets older, there's an increase in the probability that the possibility of having a cancer at an old age is higher than having it at a younger age. So this is all we have for the lesson. So the quick questions we have here: which is the reason why a cell remains small? So you can check out answers. So which describe the activities of the cell that include cellular growth. So we did this in a class. So you can also go over them yourself, find answers to them. So what is the role of the cyclin? Which substances form cyclin dependent, and also what is the characteristics of the cancer cells? So this marks all of what is apoptosis. So you can find answers yourself. So this would mark the end of all we've done. The answer should be D, but I can tick on them. So I will leave that to you to do yourself, inshallah. So I wish you all the best. So if you have any questions, please feel free to send me a question. Send me a message, and I'll be ready to answer your questions. Barakallahu fikum. Take care.